Okay, so today we'll start with steroids. Okay, steroids, cardiac glycosides, and triterpenoids. Okay, steroids, cardiac glycosides, and triterpenoids. Uh, you have already studied in the previous semester about the steroidal glycosides, cardiac glycosides, and also the triterpenoid glycoside. So, uh, I will not give very details of it. Okay. So, in your syllabus, there is the steroidal glycosides, mainly the saponin glycosides. Okay, like the liquorice and aspuria and cardiac glycoside containing drug, the digitalis. Okay. Firstly, I will start with the cardiac glycoside. অনামিত্র অনামিত্র
Yes, ma'am.
আমাকে তুমি হোস্ট হয়ে গেছো, আমাকে একটু হোস্ট করে দাও।
Okay, ma'am, করে দিচ্ছি।
ওকে, প্রত্যেকে শুনতে পাচ্ছ তো? স্ক্রিনটা দেখা যাচ্ছে?
হ্যাঁ ম্যাম।
Okay. So there are number of steroidal glycosides. There are number of steroidal glycosides or steroidal molecules which have cardioactive property. Okay. So basically the cardiac glycosides are the steroidal glycosides, and they are used in or have beneficial and effect or toxic effect on the heart. Okay, they are mainly used in treatment of congestive heart failure and also used in the treatment of arterial fibrillation. Okay, uh, so there are these are the drugs which are also known as cardiotonics. Okay, these are the drugs which are known as cardiotonics. As we all know that the main activity of the glycosides is due to the aglycol part. Okay. The main activity of the glycosides is due to the aglycol part. The aglycol part, which is a steroidal molecule, in the cardiac glycosides, and basically there are two types of cardiac glycosides. One is cardinolide and another is bufordinolide. One is cardinolide and another is bufordinolide. Both the cardinolide and bufordinolide has a lactone ring attached with them. Okay. Both the cardinolide and bufordinolide have a lactone ring attached with them. Okay. In cardinolide, the steroidal moiety is basically mainly the C24 or C23 in nature. Okay, the steroidal moiety is basically C23 or C24 in nature. Means it contains 23 carbon atoms or 24 carbon atoms. Okay, and in the cardinolides, there are C23 carbon atoms. Okay, and in case of bufordinolide, there are C24 carbon atoms. Okay, or cardinolide is known as C23 steroids having a alpha beta unsaturated 5-membered lactone ring, which is attached at the beta position, 17 beta position, which is very important because depending upon the position of the lactone ring, depending upon the position of the lactone ring, it distinguishes which type of cardiotonic glycoside is present, either cardinolide or bufordinolide. In case of bufordinolide, there is the 6-membered lactone ring present at the 17 alpha position. Okay, present at the 17 alpha position. So, in case of bufordinolide, there is C24 steroid or carbon atoms having a 6-membered lactone ring at the 17 alpha position. Whereas in cardinolides, there is C23 carbon atoms having a alpha beta unsaturated 5-membered lactone ring attached at the 17 beta position. Okay, depending upon the lactone ring position, placement of the lactone ring and the number of the 6-membered or 5-membered lactone ring, there are different types of cardiac glycoside. Okay. Next is as you can see, this is the structure of the cardinolide and this is the structure of the bufordinolide. Okay. And one more thing, the main core structure, in case of a steroidal glycoside is the cyclopentanoparhydrophenanthin tree. Okay. In case of steroidal glycosides, the main core structure is the cyclopentanoparhydrophenanthin ring. This is the cyclopentanoparhydrophenanthin ring. This is the cyclopentanoparhydrophenanthin ring containing 4 ring structure. Okay, 4 ring structure. This is the basic moiety of the steroidal glycosides, and this is known as the cyclopentanoparhydrophenanthin ring. Okay. Next is this is the lactone ring. This lactone ring is responsible for the specific cardiac activity, okay, in case of cardenolites and also in case of bufadenolite. This is the 6-membered lactone ring and this is the 5-membered lactone ring. Okay. Next is the example. Cardenolites, example is the digitalis, strophanthus, oleander. In case of bufadenolite, example is squill. Okay. Example is squill, specia. This is the example. And also the name bufadenolite is derived from the generic name of the toad or bufo. Okay. These are mainly found in the skin of toads. Okay. Scientific name is a Bufo melanostrictus. From this, the bufadenolite term has arisen, and the compound bufalene is important, which is isolated from the skin of toad. Okay. So this is the basic structure of the cardenolites and bufadenolites. Next, we'll move to the digitalis, the cardiotonic drug, the cardinolite drug, which is the digitalis. Okay. Digitalis. So basically, digitalis is a cardinolite containing C23 carbon atoms containing the lactone ring at the 17th beta position, and the lactone ring is the 5-membered lactone ring, alpha beta unsaturated 5-membered lactone ring. Okay. And this digitalis is a cardiotonic drug and it has a strong action and a powerful action on the cardiac muscle. Okay, which you have already studied in your pharmacology part. Next is the details of the digitalis. Digitalis is also known as foxglove. Digitalis is also known as foxglove. Okay. Biological source in digitalis. In case of digitalis, we normally use the digitalis leaves. The digitalis consists of the dried leaves of digitalis purpurea. Families is Chlorofluaceae, and already discussed that the main cardiac activity of the cardiotonic drugs is due to the aglycone moiety or the aglycone part. Next is there are two types of glycosides present in digitalis. One is the primary glycoside and another is the secondary glycoside. Okay. There are two types of glycosides mainly present in digitalis. One is the primary glycoside and another is the secondary glycoside. Okay. And the total cardenolites present in digitalis is not more than 0.3%, which is the digitoxin. Okay. Which is the digitoxin. One more part, that digitoxin, it is the glycoside. Digitoxin, it is the glycoside. It contains the sugar part and the non-sugar part. Okay. Digitoxin is composed of the sugar part and the non-sugar part. Here in the structure. Here is the structure. Okay. This is the sugar part, D-digitoxin. It is the sugar part which is attached at the C3 position. Okay. The sugar part is mainly attached at the C3 position. The sugar part is attached at the C3 position. Okay, this is the digitoxogenin. This is the digitoxogenin. This is the genin part, or this is the aglycon part, and this is the sugar part. So both the sugar part and the non-sugar part combinedly form the glycoside. Okay, both the sugar part and the non-sugar part combinedly form the glycoside. So while writing the glycoside, that is the digitoxin, you have to write the sugar part and the non-sugar part. Okay. These digitoxins, these are the sugar part, and this is the digitoxygenin, and gitoxygenin, it is the non-sugar part or the aglycon part. While writing the glycoside, you have to combine the sugar part and the non-sugar part. Okay, combine the sugar part and the non-sugar part. One more thing. This is the C3 part of the aglycon, but this is the C3 position of the aglycon part. This is the C3 position of the aglycon part. Here, the sugar part will be attached in the final glycoside structure. Okay. Here, this is the C3 position of the non-sugar part or the aglycon part. Here, the sugar part, this sugar part will be attached at the C3 position and will form the main glycoside. Okay. Next. So basically, digitalis is a perennial heart about 1 to 2 meter in height. Normally found in India in the hilly areas. In India, it is found in the hilly areas like the Kashmir, Nilgiri Hills, and apart from India, it is found in England, Europe, United States, etc. So basically, digitalis, it is a harp and it requires some standard conditions for cultivation. Okay. Next is the organoleptic characters. Organoleptic characters, the leaves are mainly 10 to 40 centimeter long. The leaves are mainly long, somehow longer than a normal leaves. Okay. And the shape is ovate lanceolate, and the color is dark green, dark grayish green in color. Taste is bitter. Okay. And the leaves can be identified by the presence of wrinkles in the leaves, wrinkles and veins which are depressed in nature. This can be easily identified from the other plant leaves. Okay. Uh, Next is the microscopical characters. The main microscopical characters like the it contains anemocytic stomata. This specific point is important for your GPAT preparation or competitive exam. This the microscopical characters of the specific drugs, like the which type of trichomes are present, which type of stomata are present. This is very important in case of competitive exam. So you have to take special care in these points. Okay. As you can see, the digital slips contain anemocytic stomata, different types of trichomes like the glandular trichomes, which are multicellular uniseriate in nature. There are starch grains also present. Okay, and apart from glandular trichomes, it also contains covering trichomes and calcium oxalate crystals. Next is the chemical constituent, which is very important. As already discussed that digitalis contains two types of glycoside, mainly the primary glycoside and the secondary glycoside. Here it is clearly mentioned which are the primary glycoside and which are the secondary glycoside. Apart from that, digitalis mainly contains 30 to 35 types of cardiac glycosides. Okay, 30 to 35 types of cardiac glycosides, and both present in combination with primary and secondary glycosides. Okay, and purpurea glycoside A and purpurea glycoside B are the important primary glycosides. Okay, and the sugar part is mainly the digitoxose, D-digitoxose, and digitallose. This is the sugar part, and the secondary glycosides are the digitoxin, G-toxin, gitaloxin. This is the secondary glycosides. Okay, and there is an conversion that the purpurea glycoside A and the purpurea glycoside B. The purpurea glycoside A on enzymatic hydrolysis yields the digitoxygenine, the glycon part, and 3-digitoxose molecule. Three digitoxose molecule, which is the sugar part. Similarly, purpurea glycoside B yields the digitoxygenine, the aglycon part, and the 3-digitoxose molecule, which is the sugar part. Okay, which is the sugar part. So, in the main glycoside part, there will be 3 molecules of sugar part and 1 molecule of aglycon part. Okay. And the digitoxin and the G-toxin are the main active component of the drug. Okay. Next is the apart from the primary glycoside and secondary glycoside, there are 2 supporting glycosides which are present, which is the digitonin and G-tonin. And apart from this, also contains flavonoids, fatty substances, pectin, etc. Okay, so this is the main chemical constituents of the digitalis. Digitalis is a very important drug, very important in terms of cardiac glycosides, and the sugar part which are mainly present, which I have shown earlier, the sugar part which are mainly present is the D-digitallose, and digitoxose. These 2 sugar parts are present in digitalis. Next is the chemical test. There are 3 chemical tests. One is the Keller-Kiliani test, which is confirmatory for the cardiac glycosides. Next is the legal test and Balzet test, which is confirmatory for the steroidal containing glycosides. Okay, and also cardiac glycosides. So these 3 tests is for the cardiac glycosides mainly. Okay, and in legal test, sodium nitrophosphatide is used along with pyridine, which gives pink or red color. In Balzet test, digitalis with sodium picrate shows yellow to orange color. And in the later we'll discuss about the Keller-Kiliani test. Okay. Next is the use. Digitalis is mainly used in congestive heart failure and also in other heart diseases. Okay. It mainly blocks the sodium potassium ATPase pump in the cardiac muscle and also used in arterial fibrillation. Next is the one more point, digitalis has a cumulative effect in body, and digitalis has cumulative effect in body and is slowly eliminated. Okay, cumulative effect in body and is slowly eliminated. And next is the adulterants. This adulterants is also important in case of your competitive exams. Okay, this adulterants is also important, and the adulterants which are mainly used are the leaves of Bharvascum capsus, primrose leaf, or scientific name is the Primula vulgaris, and comfrey leaf, scientific name is the Symphytum officinale. Okay, and these adulterants can be identified with the help of the microscopical characteristics. Okay, and next is the allied drugs, the related drugs. Allied drugs are the related drugs. Allied drugs Digitalis is the Digitalis lanata, Digitalis lutea, and Digitalis thapsi. Digitalis lanata, Digitalis lutea, and Digitalis thapsi. As already mentioned here, you can see that the Digitalis lanata has 70 types of cardiac leukocytes present. Digitalis lanata contains 70 types of cardiac glycosides, in which the primary glycosides are lanatocytes A, B, C, D, and E. In case of official drug, Digitalis purpurea, the primary glycosides are the purpurea glycosides A, B, C, B. And in case of Digitalis lanata, the primary glycosides are lanatocytes A, B, C, D, and E. Next is the aglycone part. In case of Digitalis lanata, the aglycone part is degoxygenin and digitoxygenin. In case of Digitalis purpurea, the aglycone part is digitoxygenin and gitoxygenin. Okay, these are the distinguished characters. And next is the Digitalis lutea. Digitalis lutea is also as potent as Digitalis purpurea. And Digitalis thapsi is more potent than Digitalis, 1.25 to 3 times more potent than Digitalis. Okay, so these are the allied drugs of Digitalis purpurea. This is the structure of the primary glycoside. Sorry, this is the structure of the glycoside part and the sugar part. Next is the Keller-Killiani test. In the Keller-Killiani test, alcohol is used along with lead acetate, and also glacial acetic acid is used. Finally, it will give, if cardiac glycoside is present, then it will give reddish brown color, or which will turn to bluish green color. And if the cardiac glycoside is present, then it will give the positive bluish green color in the end. So basically, Keller-Killiani test, legal test, and Valjet test are the confirmatory tests for the cardiac glycosides. Okay. Moving to the next section is the steroidal glycosides. Moving to the next section is the steroidal, this is the steroidal glycoside containing the saponin glycosides. So, steroidal glycosides. Steroidal. Hello? কেউ কিছু বলছো? কেউ কিছু বলছো? So, next is the saponin glycosides. Saponin glycosides, the word saponin means, the word saponin derived from the Latin word sapo, means the soap. Okay. So, these are the glycoside containing drugs which have the soap forming property or the froth forming property. Okay. Okay, this type of, as you can see, this type of glycosides contains a high percentage of like this type of drug contains a high percentage of glycosides which have the property of forming soap or froth, uh, producing a frothing aqueous solution, which has the property of producing a frothing aqueous solution. So this type of drugs contains some foam forming or the froth forming or soap-like properties. Okay, so these are known as the saponins, and these are basically the steroidal glycosides. They also contain the main core moiety of the cyclopentanoparhydrophenanthine ring, like the cardiac glycosides. Okay, this also contains the main core structure of the cyclopentanoparhydrophenanthine ring, like the cardiac glycosides. Okay. Next important point is that this saponin glycosides have the hemolytic property. These saponin glycosides have the hemolytic property, means it can cause the hemolysis of the RBC. Okay, hemolysis of the RBC. So in some cases they are highly toxic in nature. Okay, in some cases they are highly toxic in nature. Saponins are basically high molecular weight and high polarity. Okay, and their isolation is very difficult. Next is uh, the main aglycone part is known as the sapogenin. The main aglycone part is known as the sapogenin. Okay, this is the steroidal moiety, and this is the aglycone. This is the aglycone part. This is the aglycone part. This is the steroidal skeleton, and there are mainly two types of saponin glycoside. Two kinds of saponins are there. One is the steroidal, commonly the tetracyclic triterpenoids, and another is the pentacyclic triterpenoids. And one is the tetracyclic triterpenoids, and another is the pentacyclic triterpenoids. Okay, and tetracyclic and pentacyclic. This is the steroidal skeleton, which is the tetracyclic triterpenoid, and this is the pentacyclic triterpenoid. Okay, tetracyclic and this is the pentacyclic triterpenoid. Acha. One more point that you have two drugs in your syllabus. One is the diasporia and another is the liquorice. Okay, one is the diasporia and another is the liquorice. Diasporia is the tetracyclic triterpenoid, the steroidal saponin glycoside, tetracyclic in nature, while the liquorice is the pentacyclic in nature. Liquorice is the pentacyclic in nature. So next is the tetracyclic. Next is this is the main core structure, the skeleton, the cyclopentanoparhydrophenanthine ring. And next is the moving to the glycosides, the diasporia. Okay. Firstly, we discuss about the diasporia. Mainly the steroidal saponins are not widely found in nature. Moving to the diasporia. So one more point: the biogenesis of the biogenesis of steroidal saponins is mainly through the mevalonic acid pathway. From the mevalonic acid pathway, we get the squalene, and from the squalene, we get the cholesterol moiety, and from the cholesterol moiety, we get the different saponin glycosides like the diastenin. Okay. in case of diasporia from the diagenesis. So the biogenesis is through the mevalonic acid pathway. From the mevalonic acid pathway or the acetic pathway, we get the squalene molecule, and from the squalene we get the cholesterol molecule, and from the cholesterol molecule we can get easily the diagenin or the sapogenin part of the diasporia. Okay, this is the biogenesis of the steroidal glycoside. Next is the details of the diasporia species. Diasporia is also known as yam. Okay, diasporia is also known as yam. Just a minute. Okay, diasporia, which is also known as yam or ratalu. Synonym is the yam or ratalu. Please confirm. You can see the screen share now. Please confirm.
Yes, ma'am.
You can see the screen share. Okay.
Yes,
So
ma'am.
diasporia is known as the synonym is the yam or ratalu. It is the dried rhizomes, mainly the tubers or rhizomes of different species of diasporia, like the diasporia deltoida, diasporia composite, and diasporia floribunda. Okay, this is the different species of diasporia belonging to the family of the Diasporiaceae. It is also known as rheumatism root. Okay, Diasporia is also known as rheumatism root. Rheumatism root. Commonly, it is also known as yam. It is mainly found in India, in the Himalayan region, and also apart from India, it is found in North America and Mexico. It is a perennial climber. It is a perennial climber. Okay. It is a perennial climber, and it requires specific soil conditions for the propagation. Okay. Uh, I'm not getting. I'm not giving the details of its cultivation and collection. And you can see. You can refer the notes. Okay, which we give in the classroom. And next is the characteristics. The ডায়াসপোরিয়াকে বাংলাতে বন আলুও বলা হয়, ঠিক আছে? In Bengali, it is also known as bon alu. So it is somewhat like the potato which we used normally. Okay, look like potato, and the color of the plant is slightly the color of the part, not plant. The color of the yam is slightly brown, colorless. Inside it is colorless, with bitter taste, and the size may vary. Okay, and the size may vary. Okay, next is the chemical constituents. Next is the chemical constituents. Diasporia mainly contains 75% of the starch. Okay, diasporia mainly contains 75% of the starch. They are not edible. They are non-edible. They are not edible. They are non-edible. Okay, they are not edible. But diasporia can be used for the isolation of starch. Okay, diasporia can be used for the isolation of starch as it contains the maximum amount of starch in it. Okay, and they are bitter in taste. The main active constituent of diasporia is the dioxanin. The main active constituent of the diasporia is the dioxanin, which is the aglycone part, which is mainly present at the 4 to 6% in nature. This is the structure of the dioxanin. This is the structure of the diazenin. This is the glycone part, and the main glycoside is the diosine. The main glycoside is the diosine. How this diosine is formed? This diosine is the combination of the sugar part and the glycone part diosine. Okay, diosine is the combination of the sugar part and the glycone part diosine, and the sugar part will be attached in this position. 1, 2, 3, C3 position. The sugar part will be attached here. After attaching the sugar part, the molecule will become the glycoside, and it will be known as the diosine. Okay. Next is the sapogenin part is the sapogenin part is the diazenin, and apart from it, it also contains another glycoside, steroidal sapogenin and its glycoside, another which is the smilagenin and its smilagenin, and also it contains jamogenin. Okay, and it contains the rhizomes contains an enzyme which is known as sapogenase. The rhizomes contains an enzyme which is known as sapogenase, and the tubers is also rich in glycoside and phenolic compounds. Sugar, the tubers is also rich in glycosides and phenolic compounds. Okay. And it also contains an alkaloid diascorin, resin, botagenin. Okay, and also contains small amounts of hecogenin. And apart from this, contains cholesterol, sigmasterol. Okay. These are I'm showing you some species of the diasporia. These are the different species of diasporia from which we can get the diazenin, and hecogenin is also diasporia also contains some amount of hecogenin. Okay, these are the different species, and this is the biosynthesis of diazenin. This is the biosynthesis of diazenin from cholesterol given, and this is the sugar part. Okay, this is the diazenin. This is the structure of the diosine. This is the main glycoside of the diasporia. Diosine, it is the main glycoside of the diasporia. You can see this is the diazenin part, and in the C3 position, the sugar part is attached. Here, 2 molecules of rhamnose and 1 molecule of glucose is present. Okay, 2 molecules of rhamnose and 1 molecule of glucose is present. So the sugar is known as the glucoramnose. Okay, glucoramnose part or sugar part is attached with the C3 position. One water molecule will be loose. Okay, and this is the This is the structure of the diosine. This is the diazenin, which is the sapogenin and glycone part of the diasporia, and the total molecule represents the glycoside diosine. Okay. Next is the use, which is the use of diazenin. As it is rich in the sapogenin part diazenin, and the diazenin is used as a precursor for the synthesis of several corticosteroids and sex hormones and oral contraceptives. Okay, diazenin, it is used as a precursor molecule. Diazenin, it is used as a precursor molecule for the synthesis of different steroidal corticosteroids, corticosteroids, sex hormones, and contraceptives, oral contraceptives. Okay, and it is also used for the extraction of starch, extraction and isolation of starch, and it also used in also rich in vitamin B1, B2, and B6. Apart from this, these are the main uses of diazepam. Apart from this, it's also used as antispasmodic, anti-inflammatory, anti-rheumatic. Okay, and also used in treatment of rheumatic arthritis. So, diaspora is also known as rheumatic root, as it is used in treatment of rheumatic arthritis. So, diaspora, the synonym of diaspora is rheumatic root. Okay, rheumatic root. Next is the chemical test, as it contains steroidal part. So, the Lieberman-Barger test can be used for the identification purpose, and also diaspora contains the saponins, so it will also give the hemolysis test and the foam test or Troutman-Mead test. Okay, these are the chemical tests, Lieberman-Barger test and the Lieberman reaction test. Okay, if the steroid is present, if steroidal molecule is present, then it will give the green color, and specifically red color for the triterpenoids, steroid, triterpenoids, saponin. Next is the some allied drugs, some allied species, which already discussed. The allied species are the Diascorea plurimunda, Diascorea villosa, and Diascorea deltoida. These are the some allied species of Diascorea. So that's all for the Diascorea species or the Diascorea drug. Next, we'll move to liquorize. Next, we move to liquorize. So, next is the pentacyclic triterpenoid saponins. Pentacyclic triterpenoid saponins. Pentacyclic triterpenoid saponins. Okay. These pentacyclic triterpenoid saponins are very rare in nature. They are not present in. They are not present abundantly. Okay. Mm. These pentacyclic triterpenoids Pentacyclic triterpenoids are classified in three classes. One is the beta-amarin group, and second is the alpha. First is the alpha-amarin group. Second is the beta-amarin group, and third is the luteol. Okay, these three classes of pentacyclic triterpenoids are present. And here also the sapogenic part is attached with the sugar part at the C3 position, and the biosynthesis is same. And this triterpenoid can be classified into three groups that are alpha-amarin, beta-amarin, and luteol. Okay. And the liquorized root contains the main saponin glycoside, which is the glyceric acid. Main saponin glycoside, which is the glyceric acid, about 12%. Okay. Next is details. So liquorize. So liquorize is also known as in Bengali it is known as Josti Modhu. Okay, liquorize in Bengali is known as Josti Modhu. Josti Modhu. Okay, and it is a very common and traditionally used anti-diabetic in nature. Okay, liquorized root is very traditionally used as anti-diabetic, and also it is used as expectorant. It The licorice root is traditionally used as expectorant and as anti-diabetic. Okay. Next is the licorice root is the dried unpeeled roots and stolons. Licorice is the dried unpeeled roots and stolons of Glycyrrhiza glabra. Okay. Unpeeled roots and stolons of Glycyrrhiza glabra belonging to the family of the leguminaceae. Belonging to the family of the leguminaceae. It mainly contains not less than 3% of the glycerinic acid. Glycerinic acid. Glycerinic acid. Okay. And there are different varieties. One is the Glycyrrhiza typica, Glycyrrhiza glandulifera, Glycyrrhiza glandulifera, Glycyrrhiza typica. Okay, these are the different species or varieties of the licorice. Commercially available on native to Spain, England, and also found in Russia, India. There are different varieties that like the Glycyrrhiza glandulifera is the Russian licorice. Glycyrrhiza glandulifera is the Russian licorice. And the Glycyrrhiza glabra variety violets is the Persian licorice. Okay, Persian licorice. Next is the cultivation and collection. There are three varieties. Before moving to cultivation collection, one more important point: there are three different varieties of this licorice. One is the Spanish licorice, another is the Russian licorice, and third is the Persian licorice. The Spanish licorice is the Glycyrrhiza typica, Russian is the Glycyrrhiza glandulifera, and Persian is the Glycyrrhiza violets. Okay, these are the three varieties of Glycyrrhiza. Next is the cultivation and collection. And these are obtained from the wild variety. Okay, the wild variety species. Next is the macroscopical characteristics. Color is the yellowish brown or dark brown externally, yellowish brown or dark brown externally, and taste is sweet. The taste of this root is sweet, so it is known as Joshti Modhu. As honey Modhu, it is sweet in nature, so this is known as Joshti Modhu for this sweet taste. The odor is very faint and characteristic. The size of the root is maybe 20 to 50 centimeters, 2 centimeter in diameter. The fracture is fibrous and splintery. It is basically the wood. Okay, so the fracture is basically woody and splintery. So next is the microscopical characteristics. Microscopical characteristics: presence of fibers, lignified fibers, phloem and xylem vessels, and presence of stolons, teeth, parenchymatous cells. Next is the chemical constituents. Chemical constituents. This is the pentacyclic triterpenoid saponin. The main glycoside, the chief constituent is the glycyrrhizin or glycyrrhizic acid, which is a potassium and calcium salt of glycyrrhizinic acid. Glycyrrhizin, which is the potassium and calcium salt of glycyrrhizinic acid. And the sweet taste of the glycyrrhiza is due to glycyrrhizin. This is the potassium and calcium salt of glycyrrhizinic acid. Okay, and Next is the yellow color of the licorice is due to presence of flavonoids. Due to the presence of flavonoids, they also have anti-gastric effect due to presence of flavonoids, and uh, the flavonoids which are mainly present is the liquiditine, isoliquiditine, which occurs as a glycoside. So the flavonoids are occurs as flavonol glycosides, and this liquiditine and isoliquiditine is not very stable, which on drying convert to liquiditine and liquiditigenin. Okay, and isoliquiditigenin. These are the main flavonoid glycosides. And next is okay. The root also contains some sugar, volatile compounds, beta sitosterol, starch, protein, and a bitter principle which is known as the glycerine amarine. Okay, glycerine amarine. This is a bitter principle or bitter compound present in the glycerine root. Next, the Spanish licorice contains about 5 to 10% of the glycerazine. 5 to 10% of the glycerazine. Russian variety contains 10%, and the Persian variety contains 7.5 to 13% of the glycerazine, which is the main compound of the Glycyrrhiza glabra. Okay. Glycyrrhizine is the main compound of the Glycyrrhiza glabra, and the amount of glycyrrhizine varies in the three different varieties of the glycyrrhiza glabra, okay, or the licorice. It also contains asparagine and fat. And already discussed that the yellow color of the licorice is due to the presence of the flavonoids or the flavonoid glycosides like the liquiditine and isoliquiditine. Next is another point is that Indian licorice root, and the Indian licorice root contains a coumarin derivative and a methyl isoflavone. Okay, coumarin derivative and a methyl isoflavone, which is present in the Indian licorice root. Next is the allied drugs. Allied drugs of licorice is the Manchurian licorice, which is derived from or which is obtained from Glycyrrhiza uralensis. Is chocolate brown in color. Okay. Next is the pale chocolate brown in color. It is free from sugar and contains only the glycyrrhizine. This is the adulterant light drugs of Glycyrrhiza. Other species are Glycyrrhiza aspera. You can see here there are number of other species given. Pisuta, Inclata, and Glycyrrhiza aspera. These are the other allied drugs. Next is the use. Traditionally, licorice is used in treatment of used as expectorant and emulsant in different puff mixes and also used as flavoring agent due to its sweet taste. And due to the presence of the flavonoids, due to the presence of the flavonoids, licorice is also has anti-gastric effect used in treatment of peptic ulcer, and the drug is also antispasmodic due to the presence of the flavonoid glycosides, isoliquiditine, and due to the presence of the glycerinic acid, it has a mineral corticoid activity, due to the presence of the flavonoids, due to the presence of the flavonoids, it is mainly used as it has anti-gastric activity, anti-spasmodic activity, and due to the presence of glyceridinic acid, it has mineral corticoid activity, okay, and it is used in treatment of rheumatoid arthritis and different inflammation conditions of the body. So this is the important uses of liquorize. So liquorize different preparations which are used, different preparations of liquorize which are used, like the liquorize powder, powder of the unfilled drug. This also used as extract, liquorize of fluid extract, liquorize lozenges. So these are the main uses of liquorize. So we have discussed so far the diasporia, liquorize, and the digitalis, the cardiac glycosides, digitalis, the cardiac glycosides, the diasporia species, diasporia, the saponin glycoside containing drug, the tetracyclic saponin containing drug, and the pentacyclic saponin containing drug, the liquorize. So, if anyone you have any question, can ask now. কারো কোনো কোশ্চেন থাকলে এখন জিজ্ঞেস করতে পারো related to the chapters, related to the drugs which we have discussed now. কারো কোনো কোশ্চেন আছে? I guess no.